transcription, the process of copying genetic information, the process of copying genetic information from one strand of the DNA into RNA, from one strand of the DNA into RNA is termed as transcription, is termed as transcription. Yeah, generally double-stranded DNA is involved in this process, but only 3' to 5' DNA strand is responsible for the synthesis of RNA. That is, single-stranded RNA is produced during transcription. Yeah, so there are two statements. Though there is a double-stranded DNA is involved in the transcription, why only one of the two strands will be involved in the transcription mechanism means there are two reasons. So reason number one, so when it comes to the complementarity, so like both the strands will be synthesized, RNA, then double-stranded RNA is formed, and this double-stranded RNA is formed in the sense according to Chargaff rule, both strands are complement to each other, so that there is no chance for the further process, that means translation. This is one of the reasons, predicted reason. And the second thing is, if 3' 5' strand as well as 5' 3' strand, both the strands will be synthesized as two mRNA strands in the sense, and the nucleotides present in the mRNA quite different from one another. So that what happens in the sense, the nucleotides, the types are same only, 4 types, but the sequence of these nucleotides are different, hence the amino acid sequence also will be different during translation. Simultaneously, there is a chance for the formation of proteins, that is, two different kinds of proteins will be synthesized. So it would be a difficult procedure or cumbersome procedure. So that's the reason only one of the double-stranded DNA molecules will be involved. So two single-stranded DNA molecules are present. From the two single-stranded DNA molecules, only 3' to 5' DNA strand involved in the synthesis of mRNA or production of mRNA. Transcription unit. Transcription unit. So what are present in the transcription unit? Actually, a promoter, structural gene, and a terminator is present. Yes, this is one strand, this is another strand, this is 3' to 5', and 5' 3' strand, and this is what we can call it as promoter, and this is terminator. So as we mentioned, always polymerization takes place from 5' to 3' direction. Yeah, so this is a promoter, and this region is a terminator. So in between promoter and terminator, structural genes are flanked. So these are structural genes or structural gene, and this is what we can call it as coding strand or non-template strand. Coding strand or non-template strand. This is also like an antisense, and this is what we can call it as template or non-coding, or also known as like an sense strand, non-coding strand. So this is the area where the transcription can be begins. Transcription start site. Transcription start site. Yeah, well, so let us take one particular sequence. The nucleotides synthesized from the 3' to 5' DNA strand, that is mRNA, is synthesized. So the nucleotides present in the mRNA 5' 3' mRNA are copied as the nucleotides present in 5' 3' mRNA strand exactly copied as 5' 3' DNA strand except at thymine positions. Yeah, so this is 3' to 5' DNA strand. You can take A, T, G, C, A, T, G, C, again A, T, G, C, 5' 3' DNA strand. And according to Chargaff rule, the complementarity is T, A, C, G, T, A, C, G, again T, A, C, G. So this is a 5' 3' DNA strand. Yeah, so this is what we can call it as template strand. Template strand, and this is a coding strand. This is coding strand, and from this mRNA is synthesized. mRNA synthesized. The mRNA polarity is polymerization always takes place from 5' to 3' in both kind of nucleic acid, whether it is DNA or RNA. Yeah, the nucleotides present in mRNA are exactly copied as 5' to 3' DNA. Yeah, so except at thymine positions. So instead of thymine, uracil is present. Instead of thymine, uracil is present in RNA. See, U, and then U. So rest of the positions are same. That means A, C, G, again A, C, G, A, C, G. That means UACG, UACG, UACG. And except at thymine positions. Why means thymine, in place of thymine, uracil is present in RNA. So this is uracil, and then uracil. Even of course thymine also a modified form of uracil, since the DNA is evoluted from RNA. Yes, this is what we can call it as double stranded DNA molecule, and this is single stranded mRNA. Single stranded mRNA. And actually the transcription will be begins at the promoter region, and this promoter region, from this promoter region the transcription will be elongates and structural genes will be transcribed, and whenever it is reached to the terminator region, so then the transcription will be completed. And mainly the enzyme which is involved in the transcription is RNA polymerase. RNA polymerase. So this is what we can call it as DNA dependent RNA polymerase. That means based on the DNA strand, the RNA synthesis will be takes place. Hence the transcription is also known as heterocatalysis, and DNA replication is known as autocatalysis. DNA replication is known as autocatalysis. All DNA polymerase enzymes are nothing but DNA dependent DNA polymerases. All RNA polymerases are DNA dependent RNA polymerase.